Hey guys, it's Miss Carlson again. We've talked about section 7.1 that talked about the cell theory and different microscopes used to look at cells. We've also talked about section 2 of chapter 7 that covered all the different organelles that play a major role in helping the cell work as one. And we talked about their functions. And now we're going to focus on a particular organelle, the cell membrane, or also known as the plasma membrane, and how it allows things to be transported back and forth from the cell. So it's important to think of the cell kind of like a state or a country where it has its own borders, just like Missouri does. So Missouri's, we're kind of like in the middle of the United States, and we're bordered by several other states. And we will transport things to and from Missouri, in order for us to get goods, especially food, to meet the demands of the population. Well, the cell membrane also acts like a border for a cell and allows certain things in and out to respond to the demands of the cell itself, which in turn plays the greatest role in homeostasis for the cell. And every living cell does exist in a liquid environment, so the way that things are transported, they go through the cell membrane first and then can be delivered to where they need to go. So why does it matter? Well, we need homeostasis in order to live. And if the cell membrane wasn't letting some things in and some things out and keeping the body balanced, then things wouldn't function properly. And you can even take a close look at this diagram over here showing you different organs and tissues and cells working together to pass on vital nutrients throughout the body. So we're going to talk about two types of transport. The first one is passive transport, where things will move across the cell membrane without needing any energy to do so. Diffusion is one of those types. It's a net movement of particles from a high to low concentration until that concentrated area becomes balanced. Now, a concentration gradient is established when an area becomes unbalanced, and you can kind of use this diagram to visualize a high concentration and then a low concentration. Molecules can't pass freely, like I said, and they're going to move one way equally to the number moving the other way until over time they become equal on both sides, as you can see in this picture. Now, there is a term at the bottom of that picture that is very important that you understand. It's a semi-permeable membrane. So this is representing how the cell membrane works. Permeable means to let things through. Semi means we only do that part of the time. So the cell membrane is selectively or semi-permeable, allowing some things to come in and some things to go out. So it's important that you understand that. And you can see from this picture that over time, that substance on either side of that membrane is going to even itself out and reach what we call equilibrium, just meaning that things or the substances are balanced on both sides. Okay, so there's another type of diffusion known as facilitated diffusion. This is where substances are helped through the cell membrane by transport proteins. Now I wanted to show you a close-up picture of this. So we have the outside of the cell here, inside of the cell here, and this of course is our cell membrane right here, made up of proteins and phospholipids. So what's important to know about these transport proteins is that they will open up and allow pathway for these larger or more differently or chemically structured substances to come through that can't just simply pass through the membrane themselves. So because these substances cannot go straight through the membrane like other substances can, the protein will actually provide that passageway, as you see here, to allow it into the cell still. So facilitated diffusion is a passive process because it does not require energy. Those are the blanks that you're going to fill in there. Now, an example of another type of diffusion would be the diffusion of water molecules from a high to low concentration, and that is known as osmosis. So it's important to remember again, osmosis is also a passive process, and it does not require what? Does not require any energy in order for water molecules to pass through the membrane. So there's some must-know vocab for you, and this is probably the most difficult part of the chapter, is the different ways that water moves. Now, if a solution is hypotonic to the inside of the cell, this means that the water concentration is greater on the outside, water will go into the cell. If it's an isotonic solution, meaning there's the same water concentration on the outside as the inside, there will be no net water movement. Things will just be flowing back and forth happily. And lastly, hypertonic means that the water concentration is greater on the inside. So there's an over or more water on the inside than on the outside, so water will leave in order to get that happy medium. Now this is what happens to an animal cell or a plant cell in these different types of solutions. So again, if the cell is in a hypotonic solution, okay, as in this state, that means there is more water on the outside than on the inside, so that cell is going to fill up with water and it's actually going to lyse or burst. Then in an isotonic solution, as you see here, this is how the cells should look, and you can see the arrows going in and out. That means water is moving freely and equally. And then lastly, you have another not so good situation for the cell, which is being placed in a hypertonic solution when there's more water inside the cell than on the outside. This creates a situation where water is going to leave, and we call that a crenated cell. It's also important for you to kind of check out how all of this happens with a plant cell as well. It looks a little differently than it does in a red blood cell, but you can see that the cell wall is always kind of maintaining itself for the most part in these situations, but it's the inside of the cell that's going to suffer and ultimately destroy the cell. So here I want you to focus on this picture. And I really want you to try to just look on one side of it because it gets kind of confusing. So remember we said that if there is more water on the inside of the cell than on the outside, that the water is going to move out. So the type of solution that the cell is placed in is a hypertonic solution, and that water, as you see the arrow showing, is going to leave. Okay, why is this happening? It's because the amount of water molecules is less on the outside. Okay, again, water, not the other substance. It's less on the outside than on the inside, so that water will move until equilibrium is reached, meaning that they're balanced on both sides with water. Okay, now the last type of transport we're going to talk about is active transport. This is when molecules move against the gradient from low to high, and energy is needed in order for this to happen. So there are transport protein pumps that help with this process. They're capable of pumping molecules through the cell membrane, and they can form concentrated areas of molecules inside a cell, or they can also remove large amounts of waste from the cell, even if the gradient is low to high. So some examples of the molecules or ions that need to be passed on this way are calcium, potassium, and sodium. And this picture here shows you that a protein pump, again, is kind of like those grape-like looking structures in the cell membrane, at least according to the diagrams, they're usually purple. And what they're allowing to happen is these molecules to be carried through, and that protein will change shape in order to bind to the molecule and then release it on the other end, again needing energy in order to be able to do this. Okay, so when the cell wants to move large quantities of materials into or out of the cell, there are two different processes for this bulk type of transport. One is endocytosis, takes materials inward, and it basically forms a vesicle, as you see in the picture, that can be transported somewhere else in the cell. Now there are two types of endocytosis that you should be aware of. One is pinocytosis and the other is phagocytosis. Pinocytosis is more liquid type, so cell drinking, and then phagocytosis, the cell membrane actually forms little arms and pulls substances in, and they normally refer to that as cell eating. Now, exocytosis is the opposite. It removes materials from the cell within vesicles that move outward from the plasma or cell membrane, shown in this picture here. So it's just a reversal process. Again, removing the waste that the cell doesn't need and getting it out of the cell. Now here's a pretty cool picture from your book. It's showing you with the transmission electron microscope a white blood cell engulfing a damaged red blood cell. And this is a picture of phagocytosis because you can see the arms of the cell membrane reaching out and wrapping itself around that red cell in order to take it in and basically destroy it and get rid of it, because the cell doesn't want it anymore if it's not a functioning cell. So pretty neat looking picture. Okay, and that is all for today. Again, hopefully you're taking good Cornell notes. Feel free to go back and form good questions to your notes now that you have finished your large note-taking part of your note-taking process. And have a good day. I'll see you next time.